on Friday. And of course it was Friday.
No, one girl who got, oh my god.
That's disgusting. So what you do is you just like, I just brush my teeth in the shower. Just there. Honestly, it's not about one thing. You just sit through.
So what's like so like I'll study over something. I wanted to check out
That's disgusting. I played with the rest.
and I wanted to
I like.
I don't want to take an L because we have to stop.
Because I didn't know what it was.
Are you serious? How is that possible?
I thought you saw it in your room. I was like, what the?
I don't know. They both are doing better.
Because Max and I like worked out together because we were both like really pissed.
Definitely sorry.
Go afterward and I told you and it was
Teacher, like, everything's.
You still remember? Oh.
Yeah, like I didn't even answer to it. And there were 10 questions, so like there's no way I did good. Yeah, my sister literally mentioned like two of them and a movie. And I was like, okay, I made it, but like
She was like, yeah, I got it. I didn't get some half on this. Like, how does that happen?
I know. So I'm like, I knew it was gonna be bad going into it.
Same.
It was worse than I thought it was. Yeah, I like thought I would.
Number 7, I literally.
Oh
I
no,
wrote down a
I
bunch
left
of
that
random
completely.
things. Oh, I just wrote down so
started
much that
writing
maybe it would
stuff down.
confuse her and she would just start giving me blank partial.
No, I would like write stuff down and it would make no sense and I would erase it. So it's like you can tell that it was written on, but there's nothing there.
Also, the entire test though.
Find it? Yeah. Was it to the right?
Yeah.
It solves the whole problem.
I heard it from you that she said there wasn't gonna be any compound. And then there was. And I'm like, you can't say that.
She's sick though, right?
Yeah, but my friend like took notes and she literally has a note on that says no naming compound.
Well, sick.
Like, girl, you can't say that. I know.
She's
It was
Obviously. You just like the steps that you expect.
Looks like you hit me right now.
Out of your legislators.
All right, so let's start. Hope you guys had a good weekend. All right, so we're gonna start off with, I wanna recap a little bit of what we went over on Friday, right? So remember the story, the big central part, central dogma of biology, right? All right, let me try it up here. So we'll start with the video. We're gonna watch it together. We're then gonna start going into the details, more specific details from the transition from DNA to RNA. And then I'm gonna go through the story twice. So if you feel like you notice that I went over the story and came back, I did it on purpose. So let's start with the video to warm us up. Then we're gonna start drawing the big map of the story that we're trying to tell today. And then the third part today, we're gonna go actually into the individual details of how it's actually working. All right, so let's start with the video.
It grows. It is processed in several ways. As the RNA grows.
So a little background to the story. Right now, we're zoomed in, we're inside the nucleus. What we're looking at here in blue is our DNA. Normally it's double-stranded, but for a little bit of time, we have this memory, which is here in yellow. It actually has opened up the DNA to make it two single strands, this little middle region. And what you're seeing is your RNA being produced. All right, so we're at the beginning of the story.
Grows. It is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus.
All right. So that's the big story, right? Gene expression, we're gonna be hitting 3 different molecules, and we're gonna be keeping track of different compartments of it, right? So at the beginning of the story, where we start, right, where you're gonna have your information stored. Right, so location-wise, we're sitting in the nucleus. And the thing that we're keeping track of is gonna be our DNA, right? From here, we get the first step, the first process that we're keeping an eye on, which is gonna be our transcription, right? Here we have our double-stranded DNA. It's being copied, or one of the strands is being copied, and we start producing our RNA molecule. So we're going to kind of the drawing, it opens up, like that. Sorry. And your RNA molecule is being copied to one of the strands, and it's being released after it's being copied. So now we have DNA, and you have your RNA. Your RNA comes to existence inside the nucleus, where it gets made, right? And then travels, goes outside, and it can get to 2 different places. Here the story can change depending on where it's gonna go. One place where the RNA can go, or what's the RNA trying to get to, is your ribosome, right? But where do we find the ribosomes?
Rough ER.
Rough ER, that's right. So we have the rough ER. We have our rough ER. So right outside the nucleus, we have this, like that. And we should have these little studs, which would be the ribosome. We can also find the ribosomes that are just floating around in the cytoplasm, like this. They're free ribosomes, right? So you just have your ribosomes. And then you're free. Once we go from there, your RNA exists from here to here. The RNA gets translated by the ribosome to become a protein. That'll be our third part. Then we have our proteins. These can either end up inside the ER, or in some cases they'll be floating around in the cytoplasm, just like that. Last week we went into the next part, which was in the Golgi. We can have the protein, how it gets trafficked to the outer compartments. The other thing we talked about was how if you work with the free ribosome, you can actually, that free protein can come back to its core and to the nucleus, right? But for today, we're actually going to super zoom in on this process, transcription, which is just how does the DNA go into the RNA. I'm going to talk a little bit about this second step, your translation. I'm going to talk about how the code works, but we're not going to go into the details until tomorrow. Yeah. All right, so we got that our backstory. Let's open up the slides. Now here, like I said, we're going to be looking at the same story. We're just going to be adding a lot more information to it. So at the beginning, we're going back all the way to the DNA itself, right? And if we're talking about DNA, we're going to be talking about how we find it in 2 different organisms. For one, we're going to be taking bacteria, which is we're going to use to kind of represent the prokaryotes, and we're also going to be talking about eukaryotes. There, the big differences are, if you remember the prokaryote, it doesn't have membranes wrapping their DNA. The DNA is just floating around in the tissue, inside of the cell itself. And what we find there is that DNA is going to be double-stranded. Bacterial. So for this, I would say almost make a table. You have your eukaryotic DNA. Here, we're going to find it that their DNA actually, you're going to find a circular DNA. So it's going to look like a really big plasmid. Circular. It's also going to be double-stranded. When you compare it to ours, for our DNA, we do have double-stranded DNA. The only difference is for us is our DNA is actually linear. And this big difference is, it's gonna show up in the amount of DNA that we actually hold. The level of DNA that we hold compared to prokaryotes, it's magnitudes bigger. And the way that we do it is that we have a way of kind of wrapping the data even tighter than the bacteria can wrap their data around these supercoils. So they do it in their nucleoid. Sorry, guys. No.
Give me a second. There we go.
Here we go. We do it in the chromatin. That's a big one that we're gonna have to bring up. You guys feel pretty comfortable with this one? Chromatin, sorry. Chromatin, right? So in here we have a complex that's a mix of our DNA sequence like this, our double-stranded DNA, and we actually also use these things called histone. They basically have a group of 4 proteins, and they actually get wrapped around with our DNA, almost like to me, I almost think like a fishing reel, and the line kind of goes around it. They do about, I think it's like 1.7 to 2 loops around it, and I think it's around 200 base pairs that are wrapped around that protein complex. This gets kind of even tighter packed, because once you have this local coil around one of these histone, like this histone complex, if those can get wrapped into themselves, this will give you this fiber. These fibers then will loop through themselves, and you get these bigger fibers. Eventually, you get to the point of you have the whole chromatin. Yes, sir.
I know I'm going back, but when you're talking about the eukaryotic DNA, what is that like the top one, circular double
Yeah,
strand?
circular, yeah.
And then for the eukaryotic one, what is that?
Linear.
Linear? Okay.
Sorry, guys, I had too much coffee this morning. Can't spell, save my life. Double check for me. Let's see. I'm not getting that myself. Eukaryotic, double-stranded, linear, yeah. So this should be the little table you guys should kind of know. If I bring out a comparison, this is what I'm going to be asking you guys about, right? So how it's saved, double-stranded. This is normally when we talk about this in ring of viruses. Some of them you can have them single-stranded, or you can have them with the DNA. And here, how you have basically the DNA compacted. Good question. All right, so we have the histones. This will be a way that you can pack the DNA itself. And the next part of that is, for the most part, when we're talking about transcription, we're talking about that phase of the cell when you actually find the DNA something like this, where it's actually open, it's not completely shut away inside the chromosomes, it's completely packed away. And this is important because this is the first level of regulation. So what we find is that if you go through mitosis, right? Sorry, let me see. Let me go back. If we're looking at between mitosis events, right? That's when you're actually, the cell's doing its job, it's doing this whole process. It needs the DNA to be open. This is when you find your euchromatin. So in my head, the way I remember this is kind of like we're talking about files in your computer. This is like when you have an open file, right? During the time that you're going to do cell division, you want to make sure that your DNA is sent to the right cells. So the cells actually do this really super tight level of compaction. And this is when you find your like zip files, and that chromatin is super compact into itself. And your DNA is basically inaccessible, right? So in here, you can find your DNA almost in 2 different states. During, like the way I studied it, or like studied before, was in development. Basically, you have your DNA has the information for every single protein inside of you. And when your cells are picking their different fates to become different tissues or different organs, they'll basically be told to shut down different parts of them. So what they'll do is in certain regions, they'll compact them something like this. It'll be super tight. The DNA is so wrapped around that protein, you can't actually get to the DNA, so you can't turn it on, or it's not regulated. And the parts that are open, those are the ones that can go transcription. And right now we're going to talk about the level of regulation that we pick up there. All right. So the DNA, 2 forms. Euchromatid, heterochromatid. In your notes, I want you to think about them as either it's an open file or a zipped file. Does anyone feel with computer terms? Yeah? Zipped files? No? No? Back in the day, you guys, we had slow internet, we had to have zip files. You'd be there all day. All right, for the next part, we're looking at here. We looked at our DNA. We should be, we'll talk about it like compact versus our free DNA. The next part is, we're going to look at what is the code behind the DNA itself, right? So in here, we've been talking about the DNA has 4 different base pairs. Yes, sir.
I just had a question about the last one, heterochromatin, which one is a zip file and which one is?
So the heterochromatin, that's your zip file.
Oh, okay, I see.
And then your euchromatin, that's your open file. I'm
Okay.
a picture guy, so I throw them up there. I have the definitions too, but if I see the picture, I remember. Alright, so the next part is, like I said, we've been talking about DNA and the possibility that it has 4 different base pairs, right? A, T, C, G. We really haven't gotten into detail about what those base pairs means, and we're gonna a little bit dive into that today. This information is the one that's super important. It's the thing that basically our parents pass down to us. That's what tells you the proteins that you're gonna make. The proteins that you're gonna make tells you how you're gonna behave, how your body gets shaped, how you're gonna look. It's all determined from the DNA itself. And let's dive into, again, this is my second round. I'm gonna go through transcription and translation, so let's keep going. So at the beginning, we're gonna be looking at, again, we have our information right now currently stored in the DNA, double-stranded, right? From this, we go through that first process of transcription. So in this case, what we do is we use one of these sequences as a template, and we produce the RNA onto that sequence to make this RNA sequence. This RNA sequence then is almost like a copy of this top strand. You can see it doesn't have the E, now we have the U's, and it's a single-stranded chain. This is the one that takes the information that we have in the nucleus, right? Because it can travel, the RNA starts here, can get itself out to either rough or the free, the rough ER or the free ribosomes. And in here we have the next step, which is your mRNA sends the information to the next part, which is making the proteins, right? Today we're going to talk a little bit about how the code works, that from DNA to RNA, it's a 1-to-1, right? An A gets turned to an A T gets turned to a U, G to a G, 1-to-1 information change. Once we go to this next step of mRNA to the protein level, for translation, we have now a 3-to-1 code. We're using 3 different base pairs to give you the identity of a single amino acid. So now your data is going from 3 to 1. All right. So let's zoom in even more and let's see what's going on inside that sequence. Here, we're going to start with our DNA, right? This is before anything happens. You have your double-stranded DNA. I want you guys to become very comfortable with these names. So make sure you guys, these will be super bold in your notes or in the study sheet. When it's getting copied, double-stranded, it's going to open up, right? We have, I normally start from down here, but it's your template strand. That's the one that's your complementary sequence. It is organized 5' to 3'. When your polymerase is here is copying it, we're actually going to copy it. It's going to be introduced. This will be the 5' side, the first base pair. On the 3' side, we'll add the next RNA molecule. On the 3' of that, we add the next molecule. Yeah. So template strand, that's the one that's complementary with your RNA, right? Reverse complement. The other strand above, the one that's basically just opened up and not being used up, this is your coding strand. This one, when we're doing like DNA analysis, I'm going to use this because it almost looks how your transcript should be made. The only difference again is T's and U's, right? Yes, sir.
So the coding strand is the one that's being put into the RNA and then actually used, and then the template strand is the one that's not being used since RNA is only a single strand?
Correct. So it's like remember they bind in pairs, right?
Yeah.
T, C, and G. The code that we care about is this AUG, right? This is the important one. This is the one that looks just like this one. This seems to be better, but it should be a 5' side here, like this is the front of it, and this is going towards the end of it. Mm-hmm. Just like this one. This is the 5' side going towards the 3' side, right? The opposite strand that you open up in the DNA is the one that's backwards and complementary. So it doesn't code for AUG, it has TAC, which is completely different. So those 3 molecules, coding, RNA, template strands, you gotta know those. I haven't made the test yet, but it's gonna be on the test. All right, for the next step is, now that we've turned that DNA, we've pulled out this mRNA that looks like the top strand, the next part is turning it into the amino acids. So here it's very important to know that the code goes 3-to-1. You have 3 RNA molecules, right, AUG, and that gets turned into methionine. I'm not gonna make you memorize all the amino acid codes, but the one that, no matter what, you have to know is this one. ATG, that's the DNA. It gets turned to AUG, and that's the one that makes this methionine. That's the most important one, because that's the only one that can start making the amino acid chain. So in the test, for sure, that's gonna come up. You should be able to identify the different sequences and know that that's when you can start making your protein. All right, so 20 meters, there's 4 pictures. All right, so let's go a little bit into more detail about this. So when we're talking about the code, again, I keep emphasizing the code's going a 3 to 1, right, from your RNA to your protein. This is called a triplet code. When it's being produced, early on, we're trying to figure out, one of the things they thought, they didn't know if it was like, you use 3 for 1, there was any kind of overlap, but now we know it's like, no, you have to use 3 to make 1, the next 3 makes the next one. There's no like overlapping sequences between them. And this will give us the amino acid sequence. But let's get a little bit into the specific about how it works. For this part, let me see. Okay. So this is the table that we care about, or that we can use to actually identify the protein that we're making. So here, if we look at this sequence here, we start out with the AUG. Like I said, the most important one. If you use our table, right, our first letter of RNA is A, our second letter is a U, our third letter is a G. You have your AUG sequence, that's your methionine. Like I said, this is the one I expect you to memorize, for sure to know it, see it. Should be like a flag, it should just be focused on it. When you go to the next one, AUC, A, U, and C, you can see that's an isoleucine, for short, ILE. You can see that's the next amino acid. The third, UCG, same thing, U, C, G, and you have a serine, yeah. The other 3 that you should be able to identify, you have to memorize this because I will give the table, are these 3, which is the stop codons. So for these, if you find these 3 sequences, when you're translating the code, what ends up happening is that's where your amino acid chain ends. Right, so now it's turned from you have just the regular A, T, C, G sequence. Because they have these functions, like AUG, if you pick this up, start making protein, any other patterns will give you a specific amino acid. And then eventually when you're done, that code, if you find these 3 patterns, you can actually stop making the protein. So that's how you can dictate a protein, start and stop, right? Now the sequence in the middle, depending on what you use, it's gonna kind of connect back to this a little bit. So you guys remember what, we have 4 categories of amino acids, right? What's my favorite one? Amino acids. Hydroxyl, because it's what?
Like, what do you mean?
Because it's polar.
Oh, okay.
So we have the first group. We have that polar group that we care about. Where's it on the table? So it's your acidic. How do I find it? Where my OHs at? Here, OH, 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 OH. We have our polar group, right? What's the opposite of those polar hydrophilic groups? What's the other one? Right, you have this whole gang down here, the non-polar gang. And then we have these 2 different groups, right? You have your acidic and your basic. Based on those charges, on those side chains of this amino acid, it's gonna dictate how you're gonna interact with something, right? So then by setting up your DNA code to basically call upon a specific amino acid, you can make domains of a protein that have a specific function, right? Hydrophobic part, hydrophilic part, an acidic part, or a basic part. So the cool thing about the code is it lets us turn this like almost like this 2D data. That's the way I look at it, your DNA, right? 2D data into that protein. That protein gives you that 3D shape. It has all those charges, which gives you different ways of interacting. And that's where you have the function show up. When you're getting that data from DNA to the protein itself, it's very precise. It doesn't like for it to be like changed or messed up, because you could change the function of a protein. If that happens, you basically break what would be happening. So the DNA, getting from DNA to protein, is what we call, it's not ambiguous, right? So it means that if you have a specific base pair sequence, if you see an AGG, it's for sure going to give you an RNA. It's not going to be, you're not going to find that it's accidentally, or it's not going to be made into a different amino acid. Whatever it's coded for, that's the protein you're going to make. The thing is, it is redundant in the sense that if, for example, let's say it was a midterm quiz, and I give you the amino acid, right? I say serine. If you look at serine, I'll tell you, hey, tell me what DNA made it, and you have to go backwards. Here, what you're gonna see is if you use this table, if you use ACU, so we find serine, you see ACU, ACC, ACA, and too much coffee, UCG, fixed glycine. You know what I mean? Those things could be changing within themselves, so going backwards, is you could use 4 different sequences to give you the same amino acid. But a sequence is not gonna mess up and give you a different amino acid. Does that make sense? I will put that on the test. I haven't made it yet, but I like it. All right, so remember these 2 terms, redundant, but not ambiguous. Everyone clear on those? Yeah, all right. Oh, and then here's my cool story here. This code of translating RNA to DNA, it's like common across different organisms. So if you give the DNA from one organism to the next one, they'll technically still be able to translate your same code. So this is there's a video at the end, I'm gonna post it online. I want you guys to look it up. It's for diabetes, and then they basically use the DNA code from us. I believe they put it into bacteria, and they can actually make the protein insulin. And because we share the code, even though it's a prokaryote to eukaryote, they can do the work for us, right? All right. And sorry, I keep forgetting the keywords. And then for this, we have the reading frame. So this is going to be important. Like I said, the thing I want you guys to always hunt for, in this case, it's if you looked at the base pair, you just started picking them at random, you can see this UCA, which gives you serine, right? If you started at the second base pair, you have this, say, CAU, you have a histidine, but you guys won't be getting tricked if I do that to you guys in the midterm, because you guys know that nothing starts unless we have the AUG come first, yeah? But based on how you shift the pattern, you can change the amino acids after it. This is super important when it comes to why we have such fidelity with the DNA, RNA, and the protein itself. All right, now that I got off my tangent in the proteins, I'm gonna go back one more time. We're gonna go start again at the DNA, and we're gonna come out of how we produce the RNA. Is everyone feeling good so far, story-wise? Yeah? All right, so let's go for this. So we're gonna separate them a little bit about gene expression. I'm gonna talk about the hard one. You guys are gonna go back and look up the easy one. So I'm gonna go with the eukaryotes. So for us, we have 3 different types of polymerases. The only one, or the one that for now I'm gonna talk to you guys a lot about, is polymerase number 2. That's gonna be, I should have put here, RNA polymerase, so it should say pol 2. From now on, if you see an RNA polymerase, that's the one I'm talking about. And from here, we're gonna break down the different steps. In the book, they talk about 3, which is initiation, elongation, and termination. I'm gonna talk a little bit about what I call step 0, which is the prep work. But let's get into it. So here we go. So for the initiation of this, right? Where do you actually start turning the DNA into the RNA? There's actually clues in the DNA itself. So we have additional information that tells us when and where we are doing these functions. That's what I call my step 0, or your prep work. So in here, when we're looking at this DNA, right? Here we're looking at it in blue. The part that's actually going to be getting made into RNA is going to start from the starting point, going this way towards the blue section. The DNA in front of that, outside of that, which doesn't get made, this part here, this doesn't get made into RNA itself, but that's the thing that gives you information about when to do it. So for me, in my head, a promoter gives you 3 functions: when, where, and how much to make of the gene. Yeah. On this inside strand, you're going to see that the RNA starts a little bit earlier, but the information that it has is what to make. That'll be the specific DNAs, the AUG to start methionine, and whatever amino acids we have after. Yeah. All right, promoter of transcription. So promoter is going to give us that information. In order to do this, we have this thing called transcription factors. Now these guys are proteins, here shown in purple, and these proteins, they actually have regions on them that bind to the DNA itself. So you guys remember when we were looking at the double helix of the DNA, A and T kind of bind each other like this? There was extra functional groups left at the edges. Those functional groups are the ones that these proteins can go and interact with and actually find a specific match. So they'll have preferences for sequences. One of the ones that's known is these Tata boxes, TATA, and a few X-rays in the back. They'll find these regions. The protein itself has evolved to be efficient at recognizing the sequence and binds to it well. The other thing is, those sequences are also very useful because at these sites, if you're trying to separate the DNA, right, like that's your goal. C and Gs have 3 hydrogen bonds, means your DNA is more stuck to itself. T and As only have 2, so it's a little easier to separate the 2 strands. These first factors recognize where they're gonna function, as in what gene we're gonna be working with. They also help initiate this thing called the transcription initiation complex. And that's basically adding in that big player that I was talking about, the RNA polymerase 2. This is the one that's gonna be doing most of the work. So once this gets created, right, you have your promoter sequence, your transcription factor, and your polymerase, all of them have bound to this region in front of the gene you're gonna transcribe. They basically open up a little bubble of the double-stranded DNA. Once the bubble's open, the cool thing about this polymerase is that it can actually open up the strands. It has a pocket where it holds on to the other one, opens it up this way. It has ways to open up the DNA ahead of itself, as in making it single-stranded. And it can start adding RNA molecules, like the individual base pairs, matching them up. Right? It's moving through the sequence, it's going, all right, we found a T, add an A, we found an A, add a T, we found a C, add a G. Sorry, not add a T, you add a U. But you know, you add the molecules forward, and as it's moving forward, it keeps opening up the DNA, adding more molecules, and will make the chain behind it. That's the good stuff. And then the other cool thing about it is that the way it does is, like other polymerases, they actually need a place to grab on, but this one doesn't carry. It can just literally come in and throw in the first base pair, so it doesn't need a primer to bind onto to add DNA to it. So the first 5' doesn't need to be attached to any of the 3'. Just start throwing the chain right in the middle. And again, let's go over the sequences we talked about before. You have this top strand and this bottom strand. What's his name? This one? Forgot. Top strand. What do we got?
Coding.
Coding, yes, sir. How about the bottom strand?
Template.
That's right, you have the template, right? So when we're looking at those here, I actually drew the names for them, right? 5 to 3', 5 to 3' for the bottom strand. The polymerase itself, when it's adding the RNA molecules, it sits them down, the 5' is pointing this way, it leaves the 3' open. When it moves forward, it can glue the new molecule, its 5' into the 3' of the one that was right there. So when we talk about the direction that it travels, or it synthesizes, you can see it makes them from the 5' to the 3' direction. So it'll be gluing them basically going forward. So I'll ask you guys to draw this. Don't be like me. Make sure you label the top, the bottom, and give me the 5' and 3'. Yeah. So, all right, let's keep going. I guess I got a little ahead of myself. All right, so when we talk about this, the promoter sequences themselves, if you look at them compared to a lot of other, across different genes, or if you look across different species, the thing we're gonna find again is gonna be those TA, those Tata boxes. The thing I want you guys to remember is all the way to section 1, talking about these TAs being the easier molecule to rip apart. That helps make your DNA single-stranded. When it comes to the polymerase, here, like I said, for now, for eukaryotes, all I want you guys to think about is RNA polymerase 2. That's the big player for us. We have 2 other ones. There's extra details in the notes if you guys want to look up, but I'm going to bring them up later and I'll discuss them more later. For prokaryotes there, they only have 1. They use it for everything. And the other note here is, yeah, the TA is conserved, so that's fine. Okay, so I feel good with the story. Everyone feel comfortable with the TA boxes, how they're helpful? Right. Now, that was the first step, which is initiation, which, like I said, was you want to get your transcription factors to find the target we're going to work with. You want to bring in the polymerase. You want to have it on, open the RNA, the DNA, start making your RNA. That's your initiation. The next step is just elongation, which is just you keep doing the process over and over, opening DNA, adding an RNA molecule, open the DNA, add an RNA molecule. In that bubble, what I want you guys to notice is, while it's working with the polymerase, it'll be single-stranded, but in front, the area where we're going towards, it's still double-stranded until the polymerase opens it. And behind it, the DNA is kind of seeming back on itself. Because you can't have that third molecule interact, what we see is that the new main RNA is kind of getting kicked out and going towards the bottom. In a lot of movies that you'll see, they love to put 3 or 4 DNA loops together, like to talk about aliens and stuff. They always make me mad. It's like, no. Hydrogen bonds only work with 2. So if you guys see a movie, let me know, and then you'll see that I was right. Okay, so that's fine. These things, I was super impressed looking at them. So you can actually judge a polymerase based on two functions: how fast it can move forward during this elongation process, and the other one is accuracy. So for the one they talked about eukaryotes, they're doing about 40 nucleotides per second. Right? That means in 1 second they're adding 40 RNA molecules, right? Individual base pairs. For me in the lab, I remember when we worked with them, we would buy some of them, like the cheaper ones, and their error rate was like 1 in a million. If I wanted something super precise, I could buy an expensive one that would be like, the errors would be like, I can't, it's like 10 or 100 fold above that, where they wouldn't incorporate the wrong base pair. So they're super fast and they're super accurate. Again, because it's super important, because it's gonna make the protein at the end. Extension, it's gonna, oh, sorry. So sorry, let me go back to extension. So we're growing, we're making our RNA, the polymerase is going forward. And then the third step is gonna be our termination, which is now where it ends. So here again, there's gonna be a slight difference in the bacterium. The terminator sequence, it itself helps do things of detaching the polymerase and it releases the RNA. So basically your complex just falls apart. The protein falls off, RNA falls off, the DNA clamps back on itself, and you're done. In eukaryotes, our polymerase is a little bit bigger, it has a lot more protein complexes that are helping it clamp on and grab the DNA. And the other one is it helps with the unwinding, which I'll talk about later. But for us, we have this sequence, the polyadenylation signal sequence, which is this one right here. As we get to it, here I kind of draw it in pink, and we see the sequence up here. This causes first the pre-mRNA to get cut off, that gets released. Then a second set of proteins come in and actually help remove the polymerase from the RNA itself, right? And that's how you turn it off. So you have your start sequence using your promoter, then elongation, just basically the thing's just working, walking all the way through. Once it hits the second signal, it tells it to stop, starts making RNA, and the polymerase falls off. I remember I was reading a cool paper where there were some genes where they really wanted to turn on, and they do this cool mechanism where they'll root the DNA like this. So this would be a double-stranded DNA. So that the initiation sequence, your catabox, is here, and then your termination signal is here. That way, what they'll do is your polymerase is kind of on, they make the RNA, and when they get back here, they get kicked off, fall apart, and now you have all the reagents they need to reclamp on and get the genes you can make. So you actually make it like super high, like get super high levels of gene expression. But you don't have to know that, that's just a little side story. All right, so we have the 3 steps, right? Initiation, elongation, and then termination. So for this, I want you guys to feel comfortable with this is one of the diagrams. I'm gonna make another one, but I'm gonna upload it to our listening session. So feel comfortable with this initiation, right? Your promoter sequence, so your promoter recruits transcription factors, which help bring in the polymerase. That's where you start. You use the DNA code to start transcription. Then you have your elongation step, which is you literally just walking through that sequence, or the polymerase is walking through, just making the RNA molecule. And then the final step is when you hit termination, which is there's that second sequence at the back, which tells the polymerase to stop. You release the RNA, get rid of the polymerase, and now you have your DNA. Yeah? All right, that's the big stuff I wanted to go through. Give me a second, guys. I know I put it on the side. I wanted to show off to you guys because it looks super cool. So it's there. I'm trying to do a thing now where I'm gonna put my notes on the sides. If it's not on the slides, it doesn't count. You don't have to learn it. It's just something I wanted you guys to show off. So I looked up the polymerases themselves. So this in the back, you're actually looking in blue and in pink. That's the actual polymerase complex. So you have different polypeptides come together to make one big protein. And it uses different like alpha helixes, mainly alpha helixes, to break the DNA open. So you can see here's your double-stranded DNA coming in. And this region here is where you have the little bubble being formed. This is single-stranded, and this other black line going this way is the other single-stranded DNA. So it's opened up. Within this pocket, here's where you're having the actual magic happen, right? You're having the polymerase, it's like literally holding in its hand, like in my head, I think of it like 3D, right? It's holding in its hand. It brings a nucleotide, and based on the ability for it to match up with its right partner, it has all four available, right? A, T, C, G floating around, but whatever DNA sequence it finds, it has to make sure it matches up properly. Like the 2 hydrogen bonds or 3 hydrogen bonds, it won't incorporate into them. If it matches it up, it doesn't. I think it uses magnesium, and it'll glue that nucleotide into the growing RNA chain. Only if it's okay. It'll then clamps into it. It'll make the next molecule, and it kind of takes a step forward and goes on and moves on to the next part of the DNA. So it's super precise. The charges in that domain have to be super specific because the magnesium they use to kind of glue the interaction is based off one of those polar interactions, right? You have the hydrogen bonding to help hold an ion. You literally need that there, super precise in the right area to help glue this molecule and do it DNA by DNA. All right. Eukaryotes. All right, sorry guys, I was a little nervous today. We had a guest, so I spoke a little bit fast. So I actually finished like 10 minutes early. So we can either do questions, that could be one of them. If you guys have questions, we can go over questions. The other one is, we can go over again the amino acid code, like if you guys feel comfortable with the RNA to protein, we can do like a race with each other right now. Yes?
Can I ask again, what's the purpose of the TATA box? Like I know it's the like start of the sequence, but.
So the
Start of the promoter, right?
yeah, that's the whole point, to know that that's where you start.
Yeah.
Because otherwise, let's say, like if we look at a random DNA sequence super long, all right, so I'm gonna just imagine this is a really long sequence like this, right? And then in this sequence, we have the most important amino acid sequence that I always talk about, right? Let's say you have your ATG that exists like this. And randomly, it shows up again, and you have another ATG sequence that shows up like that. Technically, you could start making an amino acid here, and you could start making an amino acid here, right? The first level of regulation is sitting at that promoter level, telling us what's the protein that we actually care about. Right? Let's say that the protein we care about, this is the big one. This is what we want, the protein, the amino acids to be made. This is like I say, insulin, right? You want it there. By having your T, A, your title box up here, your polymerase knows that it has to go and clamp here and start producing this RNA, right? If you don't have this information, technically your polymerase wouldn't know where to go. If just say magically that we lost that function in life, it might go and clamp onto this part here and just start copying from the front going down. And then what would be the issue? We would start making proteins on this side. Because the code is so specific, if we get lucky, and it's literally 3 base pairs away from it like that, 3 at a time, we might get lucky and have the same amino acid to make the same protein, right? But we have to be super lucky. If it's shifted forward one, shifted backward ones, the translation code will be completely messed up, right? Like you would get the same protein, yeah.
So that's why there's a space between the title box and where it's actually transcripted. Like the space, like why would, like when you think of start point, like why is it not like, why is there a gap?
So for us, so there's 2 techniques. Bacteria, they have smaller DNAs. If I remember correctly, what they do is they put multiple genes right next to each other, and they'll literally just blast through it and sequence the whole thing. The RNAs get processed, and each one of them becomes a different polypeptide. For us, we're more precise, it's like super precise machinery. We use that extra promoter information, right? That 0 level of regulation to be like, no, we want to make sure only this part gets made, not the whole chromosome. Okay. So that spacing there, in us, if you look at our DNA versus their DNA is stuck right next to each other. Ours, we have huge gaps. Like I worked with a gene, my gene was 700 base pairs. Upstream, it had an information gap of about 3,000. Like that's where its promoter was. And downstream, it gives another about 200. So that outer information or that gap is super useful in the sense of, again, that tells you when, where, and how much to make of a thing in the middle.
Okay.
So it's super useful, it's super critical.
Okay.
I messed around with one. I literally changed 1 base pair, like outside in the promoter, and I can make stem cells like perfectly one size, 1 base pair vessel. It's like super like important information.
So kind of like, it's almost like timing in a way, like it like the gap allows it to like, okay, I think I understand.
the gap helps target the polymerase itself.
Yes.
So technically, I could,
If it was farther up or like closer back, then it would start at a different point, causing it to completely mess up.
let's let's say I wanted you, there was a protein here, I need it, and then all of a sudden I'm like, hey, I think I found a protein up here, right, it's ATG. I could come and be like, hey, I need you to clone something for me. I wanna make this protein happen. What you would do is, you can go ahead, add extra DNA here, you would purposely add a counterbox. Now you can make the polymerase clamp on, and it actually starts making this RNA.
Okay.
So you can swap them in and out. Another one. Yeah, does that make sense?
I think so.
So that gap is super important, it's positional. Yeah, positional information, when you start getting into multicellular stuff, it also starts separating identities. Like neighboring cells can be like, you do that function, I do this function, based on that outside code. Yeah.
So I had a question about like what exactly the RNA polymerase and like promoters are. Are those just proteins like carrying out the transcription process?
Yes. So the ribosome, the polymerase, yeah, that's protein. Kind of you can see the picture I was showing with the cool blue and pink, that's all protein.
Yeah.
It uses its amino acids to recognize the DNA, clamp the DNA open, hold the base pair and glue it, and it holds the RNA. It's doing all those jobs. The ribosome, this was a little bit more complex. It uses protein and RNA, but I'm gonna get into that one tomorrow, on Wednesday.
So this is just making mRNA.
Just mRNA, today just literally more notebooks than one.
Okay, and the promoter, you said that's also just a protein, or that's part of the polymerase?
I said that. So the promoter is the DNA itself,
Oh, okay.
right? So the DNA is just a sequence that you find. For this case, it'd be T18, that's the promoter sequence.
That's the start of the sequence.
That's where you signal the start. The one that actually doing the job, that's the one that comes in afterwards. And this is your transcription factor. This is the protein, right? So there's protein here. And this is DNA. This one will come, clamp onto the site, bind the DNA, and now the interaction of this protein, this one can interact with your goal-to physically, right? The sequence is what the transcription factor is going to stick to. This will bring the polymerase, throw it onto the DNA, and let it fire off. That makes sense? All right. Those are really good questions, guys. Appreciate it. So you guys want to do we have a
Sorry, this is a quick question. Are we gonna have to know a lot about the codon like reading frames? Like do we basically just need to know that UAA is like the stop codon?
those three, I want you to recognize them because again, they turn just a regular sequence into a specific function, right? ATG is just a DNA sequence, ATG. When it gets to that point, ATG says, we're gonna start making protein right here, right? The other ones is the three that you talked about at the end. Those are the other three that, it's just 3 base pairs in a row, but because of the pattern of what you have there, it says stop, and you stop making the protein. I will have you in the test, I'm telling you, I'm gonna make you translate them forwards and backwards, meaning that if I give you a DNA sequence and say, hey, this is the template strand, or if I say this is the coding strand, you then have to look at it and see whether you're gonna use the ATG, and then this gets turned, let me put that on now. Alright, so using this table, if I give you a sequence, like, that's what I was thinking, maybe we could do races, or we're gonna do them on Wednesday. But, oh no, sorry, next Wednesday. So when we do the sequence, and you have this show up, ATG, this is your RNA. AUG, CCG, and U, like this. There's our RNA, you use the 3, 1, 2, 3, and use the table like this, right? A, U, and then you see G, that's our methionine, that's our first amino acid, right? For the next one, you basically use the table again. CCG, that's a protein, right? This will give us the next amino acid. And then finally, UUU, what do I get?
Phenylalanine.
Right. And then finally, the last one we should be getting should be something like either the UAA, UAG, or the UGA to give us that stop. If I tell you the sequence stops, let's say we want to go backwards now, and I say, hey, this is the amino acid sequence that I have. Can you guess what the RNA is? Going backwards, these, the stop. If you put in UAA, that's correct. If you put in UAG, that's correct. Or if you put UGA, that's correct. Right? So I'd be able to take any of the three. That's where that 3 code of RNA going to protein, going this way, there's no mistake in it. AUG is undefined. If we go from the protein going back to the DNA, you can have multiple answers that are correct.
So we should like memorize that whole table?
I'm gonna put it up there in the test.
Oh, okay, thank God.
The only one that I think, and I want you to like recognize them from, like kind of like the hydroxyl groups, is I want this one. You better be able to hunt it down. If I show it to you in the template strand, coding strand, or RNA, if in the test I mess up, let's say I wrote it wrong, and I don't tell you template strand, coding strand for the RNA, it's up to you to tell me, I'm assuming this is a blank. This is what I expect. Then the answer is correct. Yeah? All right. I think we're gonna call it for today. Thank you guys very much. No more questions?
Are you going to court? I was. Mighty fast.
All that my transcription.
I agree. Yeah, they are. Freaking eagles skipped it too. It was 6.
I think they're bringing cabinets.
There's some.
So that's gonna be rough. I don't know how they like the time, construction,
Thank you.
classes. So let me know if you run into any problems and don't
I
hesitate to
saw
tell
that,
them, like.
I was so nervous.
You did great.
I
And
was
I
like,
thought the time for questions at the end was great, by the way, so.
no, that's like 2 minutes, like,
Mhm.
in the end, I'm always like, hey guys, it's time to do the last slide, because I got too shy.
Yeah. No, I know that feeling. I've started to try to work in more time for students to ask questions because, yeah,
The
there's
one
always
that-
questions.
Yeah, the one that I'm kind of prepping for right now, because I know they have to do essays before the midterms.
Oh, yeah.
So then right now, this one is supposed to be focused on biotechnology going to medicine, right? So when it's like, I'm trying to emphasize the diseases
Mhm.
and show how they're broken at the molecular level, so then they have a chance to be like, hey, I can just. make
Right
it
about that,
easy.
yeah. No, I thought that was all very good. So I'm gonna write up a little something and then we will grab a coffee one of these days.
Perfect, I
Okay,
appreciate it.
awesome. And yeah, again, just if there.
All right, thank you.